Hello everyone and welcome to the NPTEL course on pharmacognosy and phytochemistry. In this week, we are going to learn about a new set of compounds called as glycosides. As the name indicates, glycosides are the molecules which contain sugars. In that case, how do they differ from carbohydrates? Just let's see in this session. So, glycosides are molecules essentially containing sugar as an integral part of their moiety. But apart from sugar, they also contain a non-sugar moiety often referred to as genins. So, it is a sugar plus a non-sugar joined together by a linkage. Now, this linkage is generally an hemiacetal linkage. Now, hemiacetal linkages are actually the linkages formed between the reducing group, generally an aldehyde or ketone group of the sugars, and then addition of a hydroxy or phenolic group or an alcoholic group of an aglycone. So, this aldehydic or ketone group then adds to the especially the hydroxy or phenolic group of an aglycone to form a ether like compound. This ethers are or this etheryl linkages are referred to as hemiacetal linkages, and these compounds are called as sugar ethers. Just to exemplify here, we have a molecule called as dioscin. Dioscinin is also a saponin in nature, that means it has the ability to form soap like a foam. So, what happens here is, as you can see, you have a sugars moiety and the sugars are also linked to each other, but let's just see this terminal sugar. Now, this sugar part is linked to the genin part. Now, and the point of linkage is this particular ether linkages. These are not esters, these are ether linkages, and that's why they are referred to as sugar ethers. So, sugar plus genin is basically what we call it as glycosides. Now, interestingly, why these are called saponins you can understand here. So, this behave perfectly like a surfactant. So, I can put this part as a polar head, and this part is hydrophobic. So, this part is non-polar. So, when you have a non-polar group and a polar group, they have what is called as a surfactant like capacity, and hence dioscin is a saponin. Apart from that, you will find that the aglycone group not only maybe an alcohol, it might be a cyanohydrin, it might be something to do with a sulfur like compound or a heterocyclic compound. They join in to form a different form of linkages. Like in this case, it's an O linkages, but you can have an S linkage, N linkage as or even a C linkage as well. Now, if you see carefully, sugars as such do not exhibit any pharmacological activities. We have seen in the previous sessions the main biological activity of most of the secondary metabolites is attributed to their structures, and the structural contribution in glycoside is the genin. So, the genin is the pharmacologically active moiety of glycosides, whereas sugars have a role to play in its polarity. Now, what role does polarity play? Polarity helps in solubility, polarity helps in absorption, binding, distribution, metabolism, and so on. So, we say that the sugar part is responsible for the ADME. We also call it as the pharmacokinetic parameters, whereas the genin part is responsible for the activity or pharmacodynamic parameters. Now the sugars which get attached to the genins can be very diverse. It is not always that one particular sugar is found throughout the plant. If you see in terms of abundance, glucose is the most abundant sugar. So as a result, in glycosides also majoritarily you will find glucose as the one linking with the genins, but apart from that you might have other sugars also. This includes your rhamnose, fructose, arabinose. In some cases you might even have linkages with uronic acid derivatives such as glucuronic acid. Now properties, chemically, because they are sugar ethers, they are in terms of their solubility somewhere in polar to mid polar range. So they are soluble in water, they are soluble in alcohol, but as you go on a lipophilic side, you will see that their solubility decreases, and this also has a bearing on the number of sugars. So if it is less sugars, it might still dissolve to the extent of ethyl acetate butanol, but if you go on adding more and more sugars, their solubility increases in water and completely vanishes in organic solvent. Now these sugars, if they are reducing part, is involved in formation of hemiacetal linkages, have no ability to reduce Fehling solution. So most of these glycosides, when they are intact, that is when the genin and the sugar is fused, because their reducing end is occupied, will not be able to reduce the Fehling solution. But if you are able to hydrolyze it, in some cases you can do it by acids, in some cases you can do it by enzymes. So after hydrolysis, when the sugars are free, in that case definitely they will have the ability to reduce the Fehling solution from blue color to a reddish precipitate. Now in terms of optical activity, it is found that majority of these glycosides are levorotatory in nature. Coming to the physical attributes such as their nature, very few glycosides you will find occur in crystalline form. The presence of sugars, and as more and more sugars increases, render them somewhere as amorphous substances which form sticky residues and are hygroscopic in certain extent. In some cases they are definitely free flowing solids, colorless and non-volatile in nature. Because of the sugar's moiety, their volatility is reduced. Now the sugar ethers can be hydrolyzed. This can be hydrolyzed by enzymes, it can be hydrolyzed by acids, and also it can be hydrolyzed at elevated temperatures. Now it is observed that as compared to basic pH, an acidic pH is much better in order to cleave the ether linkage and release the component sugar. Now in most of the natural glycosides, it is seen that the type of linkages is generally a beta type. The glycosides can be classified depending on the nature. Like I said, in majority you will have a beta type, but in some cases alpha is also possible. So how do you do that, or what are this alpha and beta? So if it all depends whether your glycosidic bond lies above or below the plane. So if you see a typical alpha glucose, you can see the anomeric carbon. This is what is referred to as the anomeric carbon, and then you check the hydroxy which is located on anomeric carbon. If it is below the plane of the cyclic sugar, you call it as alpha linkage. Now in this case, let's say it etherifies with methanol. So you get a methyl ester, but because this methyl ester is below the plane of sugar, you call it as a alpha glycoside or alpha methyl glycoside sugar. Now in this case, what you see is this ether linkage with methanol is gone above the plane of sugar, especially if you see it with respect to the anomeric carbon, and you call it as a beta glycoside linkages. Now why is this alpha and beta linkages important? You will see that depending upon how the linkages are there are specific enzymes. not every enzyme can digest all type of linkages. Take for example, in your saliva you have your alpha amylase. In some cases you have maltase, and alpha amylases predominantly are known for hydrolyzing alpha linkages. That means if there are this kind of linkages in your molecule, alpha linkages, then your alpha amylase will be specifically able to digest it. But if your molecule has a beta kind of linkages, that will be resistant to your alpha amylase. Now there are some enzymes such as emulsin. They occur in plants, especially because plant contains glycosides and plants have the mechanism to hydrolyze. So this beta emulsins are kind of located in the vesicles and the glycosides are located away. When the plant is being consumed by an animal, it crushes this vesicle. As a result, this glycoside, especially your emulsin, is released, and this glycosidase will cleave the beta glucoside bond. So in this case you can release the glycone moiety just by releasing the emulsin. So this is a kind of a trick that the plants use when they want to defend themselves, and this is seen in mostly your bitter almonds or plants containing cyanogenetic glycosides. Now based on the number of sugars also you can classify your glycosides. Some glycosides just contain one sugar, they are referred to as monoside. A good example is your salicin. Now salicin is an anti-inflammatory compound which has been obtained from willow bark, and you can see here that hydroxybenzyl alcohol is attached to a sugar and it's just one sugar. Because it's just one sugar, it's referred to as monoside. Now coming to case where you have your rutin. In case of rutin, you can see that your rutin is linked to a glucose, which in turn is linked to a rhamnose. So in this case you have two sugars, and this is called as bioside or bidesmoside. Now it is observed that whenever you are carrying out hydrolysis, the hemiacetal linkage is much prone to hydrolysis as compared to the second or intersugar linkages, and as a result when you do acid hydrolysis, predominantly this bond is the one which chops off first. So rutin will hydrolyze to your quercetin and you can get the glycone on acid hydrolysis. The third case you can see is triosides. Now trioside is when you have three sugars. Now this is a case of digoxin, which is used in cardiac insufficiency, taken from digitalis plant, and you see here there is same repetition, but the three sugars which are linked to each other, and these three sugars are digitoxos. So three digitoxos are linked to each other, and such compounds are called as triosides. But these are not limiting, you might even have compounds with more than three sugar linkages. Now you can also classify glycoside based on the type of linkages. So what is the molecule which is actually being attached to the sugar? Now let's take case of salicin. We just saw it right now, and you can see here in case of salicin you will see that of your hydroxybenzyl alcohol is been attached to glucose, and the point of linkage is an oxygen. In such cases they are referred to as O-glycosides. Now coming to a second case you can see a sugar here, that's again a glucose, but this time there is no oxygen. So this point of linkage is actually a carbon. So when a point of linkage is carbon, such kind of glycosides are called as C-glycosides. A good example of this is Vitexin that you see in your figure right now. Now sometimes the sugars might be attached to compounds other than carbon and oxygen, and in this case we have sulfur. So in this case you can see to the glucose what is attached here is a sulfur moiety, and that is why you call them as S-glycoside. This is seen in your mustard, sarso. So the typical acrid aroma of your sulfur or sulfuraceous aroma of your mustard is because of synegrin, which is an S-glycosides. And in your DNA you have something called as purine bases. Now this purine bases are also attached to the sugar, but the point of attachment here is neither O nor C nor S, but it's an N. In such cases, they are called as N-glycosides. So depending upon the type of linkages, we chiefly have 4 compounds, which are your O-glycosides, C-glycosides, S-glycosides and N-glycosides. Now we can also divide the compounds or the glycoside based on the nature of glycone. Now this is the most widespread distribution because the glycone can be anything. But I've just listed a few major classes of glycosides to explain depending upon glycone how we can classify them. The first case is cardiac glycoside. As the name indicates, cardiac glycosides are actually a steroidal group we refer to as cardenolides, and the steroidal group is attached to sugar, and this class of glycosides have a very good effect on your CVS, and they've been used in congestive heart failure as well as in arrhythmias. The next class is alcoholic glycosides. In this case, it's the alcoholic moiety which has been used in formation of sugars. A good example is your salicin. Now salicin is a precursor of aspirin, so it is anti-inflammatory, antipyretic and shows analgesic effects very similar to that of aspirin. The next one are actually sweet molecules. Now if you see all of this molecule contain sugars, but in terms of their sweetness, not every glycoside is sweet. In fact, majority of glycosides also have a slightly bitter taste as opposed to a sweet taste. But there are certain glycosides like stevia which are known for imparting sweetness, and this sweetness is more than 100 times to that of your normal sucrose. So here you see in your structure is a diglycoside here and a monoglycoside here. So here the sugars are linked at different position and they are responsible for imparting sweetness. Apart from stevia side, they also rubaudioside which imparts sweetness to stevia. Now we go to the next class, and those are sulfur containing S-glycoside compounds, thioglycosides. Now thioglycosides have an interesting property, we call it as allelopathy. Allelopathy is a unique feature whereby a plant can retard the growth of adjacent shrub, weed or grass, not allowing their metabolic processes to go smoothly. As a result, the plants growing in the vicinity suffer. That is what we call it as allelopathy or mutual suffering. So they suffer and they wither out eventually. A good example of this is Sinigrin. We just saw it. It's there in your black mustard. Now apart from that, we also have phenolic glycosides. Phenolic glycosides, I'll give you an example of beta-arbutin. Beta-arbutin is nowadays used in cosmetic industries for its skin brightening effect. It also has antiseptic effects and some of them are antimicrobial, and as a result phenolics by their nature are antimicrobial because phenolics have the ability to precipitate the proteins. So they are used as urinary antiseptic agents or anti-infective agents. Now apart from that, we also have cyanogenetic glycosides. Now cyanogenetic glycosides are unique in a way they are called as biological warfare weapons. Now what happens here is when we say the emulsin enzyme has the ability. Now in this case the emulsin will break and eventually what happens from this glycoside is they release what is called as hydrocyanic acid. Now hydrocyanic acid is very toxic to the predators, and as a result the animals which are consuming them are poisoned. So cyanogenetic glycosides are actually the defense chemicals produced by plants to prevent them from consumption, and definitely hydrocyanic acid has a very unpleasant taste, so that is also something we can take account of its unpalatability. Now going on to the next ones are anthraquinones. Anthraquinone glycoside, a classic example is senoside. In senoside what you will observe is there's a dianthrone moiety. Now this dianthrone moiety can have a similar kind of structure, that is a mirror structure. In that case you can call it as a senocyte A. Now what happens here is anthraquinones have a ability to irritate the gut, and this ability to irritate the gut is responsible for its laxative as well as purgative effect. The next set of compounds are coumarin glycosides. Now coumarin glycosides have a very different activities. In fact, they dilate coronary arteries. They are known to block the calcium channels, block the coagulation pathways, and also known to act against microorganisms as anti-infective agents. A classic example is a novobiocin, an antibiotic, and this is a typical coumarin nucleus located in the novobiocin. We have two more classes. One is a flavonoid glycoside. Now flavonoid glycosides example is routine, is used in case of varicose veins, wherein the wall of your arteries and walls of your veins, in case of varicose veins, become insufficient and they lose their structure and become more permeable. So these are often referred to as vitamin P, which strengthen the blood capillaries, decrease its fragility and exhibit antioxidant effects. Now saponins, a good example is diosyn, which is saw it acts as surfactant. It's used as expectorant, anti-inflammatory, as a diuretic, urinary tract disinfectants, and because of this steroidal structure, it is also used in synthesis of steroidal hormones. Where do these glycosides occur? I'm just stating a few examples to put where all these glycosides are found. They can be found in plants, they can be found in microorganisms, they can be even found in some animals. So in terms of plants, you will find glycosides in leaves such as digitalis and senna, that is your digoxin and senocide. You will find it in bark such as willow, which is salicin, and cascara is cascaroside. You will see them occurring in seeds, mostly cyanogenetic and sulfur containing glycosides, that is your thioglycosides. In roots such as your licorice, the glycyrrhizin. Rhizomes, you will find them as in Picrorhiza and Dioscorea. In certain pods, your surfactants like your shikakai, acacia and vanilla. In flowers, Sophora is a very rich source of rutin. So Sophora flowers contains almost like 20% rutin, and in some cases latexes such as aloe. Aloe latex also shows anthraquinone glycosides like your senna, but they are monomeric in nature and they have much stronger purgative effect. So here are a few more references if you wish to read more about this set of compounds, and thank you everyone for your patient listening.